Hello everyone, welcome back to our YouTube channel. Today we are going to understand endoplasmic reticulum. As you can see here in this diagram, this is nucleus, and here you can see some continuous network of membrane. This is actually known as endoplasmic reticulum. Here you can see this is continuous network of membrane here. So this is endoplasmic reticulum, and here you can see some RER and SER. What are these? These are actually two type of endoplasmic reticulum. Here RER stand for rough endoplasmic reticulum and SER stand for smooth endoplasmic reticulum. So why this is rough and smooth? So actually on the RER, or here you can see there are some substance which is present on the surface of endoplasmic reticulum. This is actually ribosome, and because of the presence of ribosome, it gives a rough appearance to the endoplasmic reticulum. So that's why it is known as rough endoplasmic reticulum. In case of smooth endoplasmic reticulum, there is no ribosome on its surface, so it gives a smooth appearance. That's why it is known as smooth endoplasmic reticulum. And now, what is the function of endoplasmic reticulum? So here you can see the synthesis, modification and transport of what? Protein and lipid. Basically the function of endoplasmic reticulum is the synthesis, modification and transport of protein and lipid. Now which part of endoplasmic reticulum will synthesis protein and which part of endoplasmic reticulum will synthesis lipid? So as you know that ribosome mainly take part in synthesis of protein. So because of on rough endoplasmic reticulum ribosome is present, so this part of endoplasmic reticulum is going to take part in protein synthesis, and smooth endoplasmic reticulum where ribosome is absent, this part of endoplasmic reticulum is going to take part in lipid formation and synthesis. After formation of protein and lipid, these are bounded in vesicles, and from these are bounded in vesicles, and then from endoplasmic reticulum it is transported to a further process to vesicles or Golgi apparatus for its destination. Here in this diagram you can see there are two membrane of nucleus, inner and outer membrane. So outer membrane is actually continuous with endoplasmic reticulum and form this large continuous network. And here one thing which you should know, that stuff is formed inside the endoplasmic reticulum. Here you can see this inside part, this is known as lumen, and actually this network of membrane form stack like a structure which is known as cisternae in case of rough endoplasmic reticulum, and in case of smooth endoplasmic reticulum this is known as tubule. Remember this. So the stuff is formed inside the lumen of endoplasmic reticulum, and then it is bounded in vesicles, and then it is transported to Golgi, and from Golgi it is distributed to its final destination. So this is all about endoplasmic reticulum, and now we have to look at Golgi apparatus. Now we are going to understand what is Golgi. Okay, now pay attention to the Golgi apparatus. So first of all, let's see what it does. Actually, you can understand it as a distribution center. Once stuff come to the Golgi apparatus, then it will be distributed to its final destination. So you can understand it as a distribution center. Now let's see how does it look like. Actually, it is stacked membrane bounded sacs. You can see here. This is Golgi apparatus, and this is endoplasmic reticulum here, and this is the membrane of nucleus, outer membrane of nucleus, which is continuous with the endoplasmic reticulum. And now you have to understand there is two part of Golgi apparatus, cis and trans. The part or the side of Golgi apparatus which is closer to the endoplasmic reticulum is known as cis, and the part of the Golgi apparatus which is closer to the plasma membrane is known as trans. So once the stuff is formed inside the endoplasmic reticulum here in the lumen, it is then transformed in the vesicle, and then it is released to the cis part of the Golgi apparatus, and from cis part of the Golgi apparatus, once this part of the Golgi apparatus received the stuff, then it will transported to the next part from like here to here, here to here, and here to here. Then from here to it is released in the form of vesicle, and then these vesicles go and bound with the plasma membrane, and then through cytosis process it is released from the cell. So this is the process of transportation. Actually, now let's see how it modifies. Actually, Golgi apparatus simply modifies stuff by adding chemical groups such as phosphate, sulfate, and carbohydrate or carbs. So this is all about Golgi apparatus. Thank you so much for watching this video. If you found our stuff and our content helpful, then consider liking and subscribing our channel. Thank you so much again for watching our videos.